Okay.
This meeting is being recorded.
Okay, so let's begin. Hi, everyone. I hope you are doing well. It's a great pleasure to be here for our next PoLSAN seminar. And today it's my great pleasure to introduce Professor Rita de Almeida, who has undergrad, master's, and PhD in physics from Universidade Federal do Rio Grande do Sul. Rita did a postdoc at Indiana University, became a professor in Porto Alegre in 1985, and was director of the Brazilian Physical Society. Currently, Rita is a full professor at Universidade Federal do Rio Grande do Sul, member of the National Institute of Science and Technology for Complex Systems, and currently she has been focusing on complex systems, biological physics, cell migration, and bioinformatics. Today, Rita will talk about a very exciting topic, active matter, active solids, and cell migration. And yeah, Rita, you have 15 minutes and 10 minutes for questions. So once again, many thanks for kindly accepting our invitation, and the floor is yours.
Okay, I thank you very much for the opportunity to talk to this community that I should have joined before, but anyway, you are always, I mean, I'm talking with my colleague Leonardo, and he's always talking about this. So I know about what's happening. Okay, I'm going to talk the epithelial-mesenchymal transformation, which involves cell migration, active solids, and everything else, right? So it's not that I'm developing, although we have tools for studying active solids, but we are trying to use active cell migration, active matter, and active solids in order to understand epithelium-mesenchymal transformation. At the end of this talk, I'm going to mention that this can be used for digital twins, and I'm going to discuss a little bit how that can happen. But first of all, I want to thank all my collaborators. I think I didn't forget anybody, and some of them are students. One of the one of the photograph of Lars is not there, but I couldn't find one because, okay. And the others are either collaborators from abroad or from Brazil, right? Here is Leo that Pablo and Silvina really knows, okay? And I thank them because without all of them, I couldn't do anything. Okay, the epithelial-mesenchymal transformation is a transformation of the phenotype. The cell is going to be organized in an epithelium, and because of that, they have an asymmetry which is basal to the, I mean, in this direction, and they are symmetric in the plane for the planar thing, right? And they don't move. They are tight they are tight connected to their neighbors, and if they move, it's just a little bit, it's just a wiggle like this. And the mesenchymal is when the cell started to work as a mesenchymal cell that can migrate, and that happens when you have a wound and you need to close the wound. So you have an epithelium who has been wounded, then the cells must migrate to close the wound and do the things. And then after the wound is healed, then you need the reversal transformation, which is the mesenchymal to epithelial. And that happens in wound healing. But also that happens a lot in development, right? Because when the embryo is developing, they need to move the cells around and make this, okay. So that will be very important to organize the organs. And then you have also this kind of epithelium-mesenchymal transformation in cancer, because then mainly when the cancer is occurring in epithelial tissue, then if you're going to have metastasis, the metastasis should migrate for some place, and the ability of migration is characteristic of a mesenchymal phenotype, right? So, oh, I'm sorry. I don't want to do this. How do I go back? Oh my God. I click here. Okay. So what you have, the epithelium. Yeah. The epithelium is something like that, right? You see that the cells are not the same in the upper side or the back side. Usually what you have here, you can have a lumen or a hole or something, I mean a cavity. And then you could have more than one layer of the cells, but then here the example is one layer only. And then you have something which is the stroma where some mesenchymal cells are there. So this is typical an epithelium, right? They have the symmetry, the planar. If you look them from above, you have the polar symmetry, but in this axis they will be unsymmetrical. So you have apical basal polarization, and you have some molecular markers. Okay, and what you do when you want to do to model the things, you use a vertex model, you use elasticity theory, you use active solids, right? So you talk about what happens, what deformation, what you see in the cells if they move one around, but they don't really exchange neighbors around here, right? They can maybe vibrate and have some wavy things, but they are not really a liquid. So what you can talk is about resistance, elasticity, constant renovation, and things like that, right? So here you have a human skin model. Here you have, this is a skin model. I mean, this is a biological thing that you grow in the lab, trying to reproduce a human skin, and here you have the human skin. Here you have the epithelium. Here you have the stroma, which is some lots and lots of extracellular matrix, and here you have something that are cells that are dying, right? You are going to get rid of them because the epithelium is constantly renovating itself. Now, okay, this is typical. If you see, I didn't have the movie, but maybe you can see all the little arrows that you see that the thing is moving something. Yeah, they are moving like that. You can study an epithelium just like that, right? So you have something that a model, when you want to try, the vertex model is something that you treat the vertices, because of that, this is the vertex model, right? And then you have some elastic constant between the vertices, and you can define cells. So for the ith cell, you can talk about the area and the perimeter of the cell, and they have what they study is just like what are the movement, the possible movement that you have in this model, right? So what you have here is something that preserves the area for a target area here, that you have a cost if you have a higher perimeter, and then you have something that has to do with moving them like that. So this is the interaction between the cells as well. And here, I'm sorry, what happens, this is typical of vertex or active matter, right? What happens if you stretch the system like that? What is the answer of the thing? If they are going to stretch like that as well, or if they are going to go go thinner, So this is different properties that the elastic solids can have, and depending on the epithelium that you have, in this case, it's this that happen. If you stretch them like that, what you're going to see is that all the thing is going to go up, because it's, remember, you are looking at them from above, right? So, but you still have a third dimension, so they can be squeezed or not, right? If you have the mesenchymal cells, what you have is migration. They can have chemotaxis. They adhere much more to the matrix component below them with the fibers that are there. And the polarization that they have is rear front, which you see like that. So if you are going to have a mesenchymal epithelium transformation, what you need is a change of of um, a change of the phenotype caused by the production of the certain molecules that are there, so the transcription will be different. That will cause the rear front polarization and will cause a completely different phenotype, right? So what you have here is what you see, even when you go to the migration, you see here some cells that are migrating in a given way, and these ones are different. Look at this one. This is slightly, I mean, it's not the same kind of movement that you can see. Here is very, you see? And here you have an intermittent case, right? So it's like that. So from a physicist point of view, I have to, you have to differentiate the cells by their behavior or by something that you can measure. I mean, one thing is I go and I look to the proteins they are transcripting, they are producing at that time, and then I'm going to characterize the phenotype by the molecular markers they have. And the other thing is when I just look to their behavior and I want to understand their behavior. In fact, this is one of the bottlenecks that you have. How do you explain the phenotype, the behavior, by explaining what is being measurements that they have? Because I see a different profile of transcription. How can I translate this into a physical measurable that I see in the cells? So the first thing that you need is to know how to measure the behavior you have. Now we decided that we could contribute by analyzing what's happening with the migration of a single cell in the beginning, just to understand what is the phenotype and how the phenotype are changing. The classical model, cartoon model for, or I mean, you can understand a lot, but it's here, it's a proposition. It has been proposed by Abercrombie in 1970s, 1980s, something like that. And what happens is that you have all these actin fibers here, which will make what we call the lamellipodium, and you have some focal adhesions to the substrate, right? And so what happens is that you can see my pointer, right? Okay, you see all these mesh, this actin mesh, these actin fibers that you have there. They organize themselves in the front because you have polarization front here, rear, and then it's going to stretch the membrane and it's go up, right? So here you have the lamellipodium, and because they are going to go down, what you see, you have a new adhesion point, and then because they are stretched and you have the cortex in the cell. The stretch is going to, push or pull the rear in the front, and this pull is going to make the adhesion to disconnect, and so it's going to go there, right? So it's not, they walk as a mammal, right? Like one point here and one something. It's just that they are rolling. It's more like a treadmill than really the way larger animals walk, right? So sorry, no, this is the only. Okay, so what we did, it was a three-dimensional model for this kind of movement, and we have a cell here. This is just it's a three-dimensional, but this is just has a vertical cut here, and we consider the yellow as being the nucleus, then the cytoplasm, and then the lamellipodium. This is a POTS model simulation, right? So it is in a grid, and then each one of the cells of each one of the sites of the grid of the POTS model, it's going to interact with their neighbors. It's going to try to maintain the area of each one of the compartments. And I can change the interaction between different types of sites. For example, this purple one is going to interact with medium, which is the black, with a given number, and interact with the yellow with another number, and with the green one is another one. The trick here to make them you see, then what we have here is that we have this, additionally, we have a NAF, what we call a NAF actin field, right? Because it's this actin is just trying to simulate this push that you have for the actin here, the push that you see here. So what we did was the sites that belong, that are of the type of the lamellipodium simulation, they produce an actin field that will rapidly decay when they are out, right? And we put a term that the probability that this site is going to be copied over the medium is much larger if you have this kind of the thing, right? If you see that the F-actin is large here and small in the medium, then the probability that the green is going to be copied over the black it will be increased. But you remember they have to maintain the air volume, each one of the compartments, and because it has, if it was in the target volume, now they have a volume which is larger than the target, and it will increase the probability that the purple is going to grow over the green. And because in this case now the purple is going to be larger than it should, then you increase the probability that the medium is going to grow into the cytoplasm. And that creates a polarization axis. That depends on this, of course, that depends on the current state of the cells. And this is true. The cell is not going to walk in everything, in every condition. But if you increase this condition, then it can start growing and then maintain this kind of polarization of movement for such a. Now this is the same cell, the simulation of the same cell. It's just seen from different perspectives. You can see here that you have the nucleus there. The nucleus, the energy between nucleus and the median is really high, so it's going to avoid the median because of that, right? So you have these cells. What we did with that was to analyze the movement, right? So when you analyze a random movement, what you do is just you calculate the mean square displacement, which here we denote by rho and not by r, because we are defining using the okay. What we did was to assume that this thing is going to be ruled by a Langevin equation. And if you have a Langevin equation, you're going to have some time, which is the persistent time, and you're going to have something which is the diffusion coefficient, right? And what you can see is that you can rewrite the Langevin equation by dividing by an adequate natural unit that will be written in terms of these parameters of the Langevin equation. Then you have I would say that a normalized or a mean square displacement that is written in terms of natural units. When you do that, right, you can see that all our simulations changing the parameters are going to overlap over it, but for the initial, or not initial, for the small displacement values, which are here. The dashed line here is exactly the solution for the Langevin equation, right? For the Langevin equation here. So you have some displacement, some differential behavior for small behavior, for small displacements, and this will going to happen for different kinds of different. I mean, phi is the fraction of sites that belong to the lamellipodium. So you have here cells with a very small lamellipodium, and then you have larger fractions of the cells that's going to be more lamellipodium. And you see that they all collapse over the same curve, but for the behavior in this small displacement thing. What happens with our cells is that what they do is they do go from here to here, right? So this is a sketch of our explanation. What is happening is that it goes from here to here, but it goes like that with many different, sorry. It goes like that. I have to. It goes without, doing like this, and then they go there. They have a preferential orientation that they are keeping there, and then but along that size they wiggle around that, right? This wiggling, this fluctuation is very short, and it's short in time, and it's short in displacement. So what you are going to see that for small times you are going to see something which is a deviation that instead of being, in this part that is you have the largest inclination, what you see here is something that is ballistic-like, not exactly ballistic, but ballistic-like here. And then you don't have ballistic anymore, which would be the case of the Langevin equation, right? What you have here is something that is approaching diffusive behavior. So if you just looked for very small time interval, you see diffusion here, right? For very small ones. If you have to have a larger picture to understand that you can have something that is ballistic, like, and if you really see in a very large scale, then you are going to have a run and walk again. So you have a Brownian motion, a ballistic, and a run and walk here, right? So instead of having two regimes as in the Langevin equation, what you see is that we have 3 dimensions in 3 regions in this behavior. And we could also calculate the probability, the average velocity, or not velocity, but medium velocity, because we have to be careful. You cannot define it in a really diffusive behavior. You cannot define velocity, right? Because the displacement would go as delta r, and the time is going as t squared, and then if you define and t goes to 0, this goes to infinity. But you can always measure the displacement for a finite time. And then if you divide this displacement for a finite time, in that time, for time in a small interval that is delta, you see if you decrease the time interval, this average velocity is going to diverge. And this is what you expect for a diffusive regime up to very, up to, time interval going to 0. That's what you do. And this was a long, a very heated discussion in the beginning of the 20th century, right? Because when Einstein just showed that diffusion had the things were diffusive, the behavior were diffusive, and if you cannot define for a really diffusion, a fully developed, I don't know how I'm going to call you, but the diffusion that keeps being diffusive, even for very small time intervals, that you cannot define velocity. Then if you cannot define velocity, you cannot define acceleration, right? And if you cannot define acceleration, there is no Newtonian law there. You see, and how we are going to do it. And remember, this is 1905. That means that you didn't know anything about quantum mechanics about this time. So the classical dynamics should work for everybody, for all the systems. And then it was a very heated discussion on how to do this until in 1908. And then after that, some other developments. Langevin just came with its equation, proposed and showed that, okay, what you have is a persistent. You can talk of the velocity between two, for example, for a gas, right? Between two collisions. In that time, the behavior is going to be ballistic. Then you have the something that will obey the Newtonian mechanics. Then you have the collisions that will make at very large time scales, you are going to see as a diffusion. But for very small time scales, velocity would be well defined, and then you can apply mechanic. But what we are seeing here is after they have attained this region where it would be ballistic-like, then it goes back to diffusion. And then and cells should, right, somehow obey ballistic dynamics. But the point is, cells are not a particle. Cells are large, extended things, and they can have lots of fluctuations in the molecular thing, and this is what happens to the lamellipodium, right? And the sum of these things that are happening almost simultaneously, they can sum up to well, something that is probably Gaussian distribution, and then you really have this this stochastic term up to the very small time intervals, right? So what you should do, okay, and also in the most interesting thing is that after we did this simulation, we also analyzed some experimental data. And what we saw is that also the simulation, the experimental data, they present this kind of deviation. And this is a measure of the fluctuations in the membrane, right? Okay, so then we put some chemotaxis because we need chemotaxis in this, oh, I'm sorry. Here you have an arrow that my student want to put in a very, something very subtle, I don't know, but too subtle in my case. Here you have a chemotaxis from one side and from the other side. So in this model, we can produce this chemotaxis that we need to describe the epithelium-mesenchymal transition if you want to do the wound healing assay, right? Okay, so back to epithelium-mesenchymal transition. You have the gene expression analysis. You should look to EMT and cell cycle because cell cycle is an important, the time scale of cell cycle is smaller than the time scale for EMT and the wound closure. So during the EMT, you are going to see that you may happen to have some cell cycle. And then we are going to use another tool that our lab created with our transcriptograms, right? So we have now the validation parameters. This is part of our project. We are constructing each step of this project, right? It's still under. And then we'll have a model for gene expression and dynamics for EMT and cell cycle. Then we incorporate, the idea is to incorporate these gene dynamics into the CompuCell that I just showed you. Then we'll have CompuCell 3D models to describe both wound healing in vitro and spheroids. Spheroids are an experiment that you do in 3 dimensions. You use cancer cells, and some of the cells you have the spheroid, and then some cancer cells will just, have either some fingers or they will escape the spheroid. And then these are the experiments that we are wanting to do that. And we are going to use original data of spheroids in a collaboration with Young Yan, sorry, from Georgia University. The wound healing, for the moment, we are going to use the literature because we didn't have any compensation. Okay, so I have to talk to you about these regulatory gene networks and epithelium-mesenchymal transition. This is a collaboration with the bioinformatics post-graduation program in Universidade do Rio Grande do Norte, north, not south, but anyway, we are collaborating. And so there are some things that I'd like to discuss is that cell life is a manifestation of the biochemical reaction that are there, and they involve lots and lots of players, right? Proteins, RNAs, and metabolites. They are essentially non-linear reactions, okay? And they have lots of interactions with the environment. Okay. We'll have lots of feedback loops and dynamically, if you want to write, if you could write all these biochemical reaction equations for them, you would find many attractors, right? So you have this possibility of this whole equation, and then you have many attractors. That means that this the solution or the result of this biochemical equation can have homeostasis or some metastable states that are intrinsically different from one another. And this has to be like that, because your cells, your liver cells is completely different from your brain cells or from your skin cells. And they all have the same homeostasis. Then what happens in attractors, right? And it's external stimuli that may trigger the transition between attractors. So this is the idea that we have been working with to deal with epithelium-mesenchymal transition. What you have two different attractors, and you want to understand what kind of simulations you're going to do. There is a very, very interesting work by Tripathi, Kessler, and Herbert Levine. It starts with in 2020, but you have some very nice results also in 2023. And they have proposed that biological networks are minimally frustrated. And the experiments they have done is just they induce an epithelium-mesenchymal transition. And they have collected the cells at different stages of this transition, and they have made the RNA-seq measurements for lots of cells, for single cells. They can do it for single cells now. This is a very interesting thing. And what they do is that they can measure for each cell the amount of RNA, messenger RNA, mRNA, right, that are being produced at a given time, that's living there. And then with that, you can make a profile of the proteins that are going to be produced in the next time, okay, some time ahead. But at that time, I mean, the messenger is already there. And what you see is that simple models for these things work, different models work, and there are Boolean models. What they did, that's what more or less they did. And for physicists, or we just love spin systems, right? So what in Herb Levine is a physicist, so he just said that the state of a given, that would be a given gene that is going to be translated, something like that. It will be +1, -1, and SI, and it depends if the energy that is going is larger than 0, is less than 0, or if it's equal to 0, right? Random networks, GIJ may assume the values +1, -1, and 0, and are randomly chosen. And comparison with biological systems with the same, what happens to the random system and what's happening in the biological system, right? Saying that now if a given gene is being produced or a given gene is not being produced, it will be 0 or 1. And stable states in biological networks have basins, large basins of attraction, and the stable states are minimally frustrated. That's what they are looking at. You see that if this is true, then it means if this is true, necessarily stable states in necessarily the biological networks will have will be the stable states of these networks will be minimally frustrated. We propose a slightly different model based on a model that we developed in 2000, some 20 years ago, well, we developed both for the immune system and for the neural network system. And what we considered is that, you see here, you have a cube in 3 dimensions, because I cannot design, I cannot draw a cube in many more dimensions, but for the example, you can see. You see that all the integers of this from 0 to 7 can be mapped to the vertices of a cube. And if I want to a larger number, I just increase the dimension of this hypercube. And then I'm going to have all the possible states of a given network of a given system of n or 2 to m 2 to m 2 to m I mean it's an integer, right? The integer from 0 to 2 to m, then you can treat all these points here as the integers, right? So what we did was to assign for each one of these points an intensity of expression of information sigma. So I'm mapping each information with an integer. And then I can define all these situations here, saying that the activity will be the sum of the y's and the average of this f tilde here, right? And then you have all the correlations that you can do. And then you can assume that in this hypercube, right, the dynamics is given by a logistic map where the term or the value of the constant, which is not constant, of the parameter of the thing, you see here, you have the logistic map. And here you have the parameter that will depend on the occupation of this information, right? And see here, if you have something that you have a y sigma that is connected to other numbers here, this is the connection between them, then you can increase lambda and from 0 to some other value, and that means that it's going to be stable in the logistic map, but you can destabilize this and you can put it to 0 as a, right? And then this xy has different values of k, depending if they are memory or they are void, right? And you do something like that. And you see that if you start from a random place, these are different, they overlap with different memories. They can remember, then forget, they can remember and stay. And if you put some temperature, then you can do something that is concatenated. You remember something, that then you remember another, then you remember another, and then another. You just do this concatenated thing. That's what we are proposing to treat these Boolean networks as proposed by her. And we are really in the beginning of the things. And just to see that we can really describe this. Now, how do we harvest the information from cells, right? The first thing is that we have a single cell RNA-seq for EMT, data on gene expression. Then we analyze with transcriptograms. The EMT, just one moment, because my computer is going to, I forgot to put it. I'm sorry. I forgot to. Sorry. That's one. Now, EMT, we have this EMT, I have data on gene expression. The EMT, the transition epithelium-mesenchymal, has been inducted by TGF-beta, which is the addition of this molecule in the culture that you are doing the experiments. And they have measured in T equals 0, T equal 1, T equal 8, T40, and some 3 other states. And Now we use transcriptogram to describe these hybrid states. This is part of the collaboration with Natal in Rio Grande do Norte, and it's part of the master thesis of my student there, Odilon Júlio. Okay, and what he did was to use what we have developed, which is we took all the genes, all the proteins that an organism has, and then we retrieved information from a string database about the protein-protein interaction. And if they interact with a given confidence score, we just keep it. If they don't interact with anything, we just neglect these genes, right? So typically from 30,000, we stay with half of them for a confidence score of 80%, which is reasonable. Then we order all these genes such that using a Monte Carlo thing is just ordering, it's just a way of putting this adjacency matrix as diagonal as I can, but I have to pay some price for the fact that we have many interactions between many genes. I'm sure you are all scientists. I'm sure that when you're about 7 years old and you had all these color pencils, you try to order by similarity. Okay, we start with the yellow, then the orange, then the red, and then you didn't know if you would go to the red for the purple or for the brown, because this is what happens. You cannot embed there. So we have some frustration to do this, and this has to do with some parameters in this ordering. But we had some ideas in how to choose. It's not perfect, but it works. In the sense you see here, you have we have plotted these profiles, saying that okay, I take some of the goal or pathway that you have listed in some place, and then you say, for example, the processing of mRNA, right? All the genes that are processed here, they are in this region. All these genes that are related sorry, to regulation of small GTPase is this dark blue here. All these and so on. And you see that you have localized you have ordered in such a way that you can localize the genes that are doing the same biological function. And this is very important. What we did now is to project the data of RNA here. Amount of each gene is being translated at that given moment. And we do a running window average. Because when I do this, I decrease noise. And this is very, very, very important for any RNA-seq. There are lots of stochastic noise, and you want to get rid of it to be able to have some measurement. Okay. This we applied for many things, many organisms, and we could only even find something that gave us some international patents for treatment, because when we applied, right? But we have done it for EMT. And when you do it for EMT, you're treating some RNA-seq single cell. You have thousands and thousands of cells that are being treated at the same time. So what you do is a principal component analysis. And what you see is that you try to make sense. Here, you remember we have measured this, the group of Herbert Levine, they have measured the cells treated and non-treated, non-treated and treated in different ways in order to induce the EMT. So you have day 0, day 1, 2, 3, 4, and 8 days. So, and you have bicolor here. And you see that you first have the blues, and at the end you have the red one, right? For some reason, you see here I give you well, when you have a component, right, you have all these things plotted in all the intensity of translation of each one of the genes that you have there, the transcriptogram. Then you rotate in order to find the dimensions or the axis where these lots of points there, where they are really varying. And that means that suppose I have something that are many points here, but they are all contained more or less in a flat thing. If I just rotate the things in order to get the axis of this plane that englobes all the points, then I can forget all the other dimensions. And I'm talking about 14,000 dimensions. So if I can tell you that you can analyze only with 6 or 7 dimensions, this is really important to be able to analyze. So the first thing you do is just see what happens for the first axis, which you call principal component 1, principal component 2. So everybody here is principal component 1. And then I just plotted the components, the coefficients of the principal component 2 as a function of the coefficients of the principal component 1. And that shows you that as you follow the time from 0 to 1 to 8 days, what you see is that you can have as a proxy of time the coefficient of the first component. When you do this, you have a transcriptogram for the principal component 1, and you can analyze what the things in respect to the center of mass of all these points, where the things are varying. So you see that there are a great depression of all these things. So principal component 1, when positive, they say they are not doing lots of these green things, which is related to energy metabolism. They are also depressed in translation, so they are not producing a lot of translation or protein things. But they have something which is here, right? So when you have here, you have the TGF-beta signaling, JAK-STAT signaling, and these things will mean that the cell fate is changing, which is, in a nutshell, very fast, is correlated to this thing. We also made a movie, but I think I'm running out of the time, so I'm going to speed up here. And so we have the metabolic pathways and EMT, we have already. So I have the epithelium, I can simulate the passive thing. The active solid is on progress because I have to take this, all the cells that I wrote in that way. And then I have to understand what are the elastic behavior of them. And then I have mesenchymal cells, collective migration. We are trying to do this. This is really a little bit complicated. We have already volume exclusion. Cell-cell adhesions we have already. External signaling and chemotaxis we have already. Cell-cell signaling we haven't put it there, right? So the mesenchymal cells isolated migration we have already described, and we have these things already, right? Many cells, you see, they avoid each other. This is the thing that we had. Now we have to put them together again. So we have already the epithelium, we have already the fully mesenchymal. Now we are working with the thing, and we can describe what's happening because we use the information from the EMT transcriptograms. Oh. So this is what we are trying to do, which is here you have wound healing assay that has been, cut here, the epithelium. So we can already describe here. We are studying the phenotype transition because we know what has to do with the information with the transcriptograms. And here you have these isolated cells just migrating that we have already done. What are the possible things, the potential possibilities? I'm just saying the same thing, but we are trying to use these in digital twins, which is precision personalized medicine, and eventually to try to use this model for therapy and drugs and tissue engineering. And thank you. I don't know how long I took.
Thank you very much, Rita.
Thank you.
Good timing. So we have now time for questions. I have a couple of questions myself, but let me wait for others to start maybe. If not, let me do one first. So you showed now at the end the POTS model for several cells. And I was wondering about whether it would be possible to do aggregation kinetics questions.
Yes.
In this spirit of what we did with Emmanuel for his thesis, which you analyzed. What do you think?
Yeah. Yes, we can. We just didn't have a time for that. We are in need of more students, Pablo.
Yeah.
This is the point, right?
Sure.
But yes, we can. We can do if they aggregate and then they aggregate, and because there are different ways that you could reconstruct the model, right? You can divide the cell, and then you're going to fill all space, then they are going to change, or they could aggregate. And this could represent different phenomena in biology. I used to say, to tell this joke all the time that I'm repeating. If you have already listened, I'm sorry. But if you have 3 hypotheses for explaining a biological phenomenon, all 5 are going to happen. Right? Because in different cases, you can have different cases and they do happen, right? Yeah, we can. And you have to, all the possibilities, because it's lots of time for natural selection to try all the possibilities. So if they are possible, they happen in some phenomenon.
That's great. I know that Silvina has a question.
Oh, thank you.
Yes.
Really very, very interesting. Then I will try to get you to talk also to collaborators of mine in Buenos Aires who do cell migration experiments and have a very interesting model of a cell implantation but anyway, that for another time.
Okay.
Then we can communicate.
Okay, but I'd like to know the references.
Yes, sure.
Yes.
What I wanted to ask you want to integrate in a model from gene expression to the dynamics of computing the migration. Now, these things occur on very different timescales. Plus, for example, the data that you show on gene expression, the level of detail that you have is very coarse-grained over time, in the sense that we're showing one day, day 2, something like that. So I wanted to ask you how you are doing to deal with these very different timescales, and also to the different level of precision with which you have that, because the cell migration you have I don't know, with a lot of detail how things move, and with the with the gene expression, you don't have that much detail. So how are you handling that, the different time scales?
Okay. This is a physicist question, right? This is really a physicist question. Yeah.
No, I mean you
And,
you want to integrate everything in a model, and
yes.
and because
Yes.
things occur on very different time scales, and I'm wondering if you can work like with separate things that then, or you need to have them connected. I really don't know. Yeah.
Okay, so what we try to do, Silvina, is to treat each problem in a different, for a thesis of a student or for a paper that I'm doing, I'm treating only one of problem. For example, only single cell migration and looking to the kinetics of the things, right? And try to give a reasonable explanation from that point of view. Then we have the transcriptograms. The transcriptograms was the idea was to increase the power of the tests when measuring the profile in the mRNA production. Then we treat all these things, but of course they are linked, right? So the idea in choosing these things that I'm focusing in one point or one phenotype or one thing, I'm just choosing which of them could contribute to this project of describing in many levels and many scales the EMT production. But when you do the study, we just do the study in that scale, right? And we use any literature that we have to help us. But you're right. But the point is, one thing, I mean, if you want to produce to have a product that you can help the biologists or the medicine or something like that, you have to integrate, right, in that, in such a way that you could, maybe not perfectly, but better than what you have already, right? Improve the results in order to be useful. So the idea is if you have technology, not only science. I mean, this science you have to do just as you were saying, just focus in something. And that's what we do in our problem. But in order to choose the different problems, then I am deciding by the idea of the product that I want to finalize, to realize. That is the point. I don't know if I have more or less answered.
Yes. Thank you. Thank you.
Okay.
I have another question, if I may,
Okay.
about the time it takes for the transition to occur. Because you mentioned that there is the epithelium state, and then you can, let's say, remove the cell from the tissue and put it isolated, and then it will express a different state.
Yes.
How long does it take for this to occur?
Yeah. About 8 days. The experiment took 8 days, right? So they have done in two batches. One, they have day 0, 1, 2, and 3, right? And then they remade the batch, just day 0 and day 4, 6, and 8. Day 0 and 4 and 8, because it's 4 and 8, right? And that happened because they underestimate the time that it's going to make some differences in the phenotype. So the idea is it's about 10 days. And if you look here, I think it's the movie, because I didn't show the movie. My students sent me, but I couldn't. Okay. So you see here is the time of the thing, right? And if it's almost zero here, the transcriptogram, it means that it is in the average. And then it will start to make difference only very in the end. So it's the last days. I mean, 7 to 8 days, that is going to be the difference of the thing. And then they go to the average. Here they are at the average. Then it goes down, and then it stops. Here is mesenchymal, right? They have these things, and this started again. So this is the epithelial thing,
Yeah, so it's
right? You
kind
can
of
see where
slow, right? It's a kind of slow.
slow. Yeah,
I mean,
yeah,
you
yeah.
cannot
So
use that.
and
Yeah. Sorry, go on.
because of that, you have to consider cell cycle as well,
Yeah.
because the cell, there will be some, and it has to, right? Because if it's wound healing, you want the cell cycle to start and to start to reproduce, to fill all the space that is there. So
Yeah,
they
so the
need
migration.
to have some cell cycle.
Yeah, so for instance, in wound healing, migration
Yeah,
can happen much faster through other means, but if you just let the cell there, it will take a long time to become migratory
yeah,
or something.
yeah, that's it. Yeah, well, in cornea, for example, cornea is going to heal for a mild cornea. You have the epithelium, and then you have stroma and some funny things happening because you have to keep the transparency of the cornea, right? But if you have a mild thing that only get the epithelium, it will heal in 2 days, right?
Mm-hmm.
But this is because it's only epithelium. The epithelium is going to, it's not really a mesenchymal thing, epithelium-mesenchymal thing. It's very mild. So you have some healings that will happen without really finalizing the EMT.
I see. I see.
Okay. So because of that, they probably are faster.
Yeah. And even before that, there is a even faster response, but not of the tissue cells, but of, I don't know, white cells that go to that wound.
Yeah,
Otherwise, it
yeah.
would be invaded by bacteria immediately, right?
Yes, well, it depends on the case, right? Because I have been studying cornea, I'm going to give you the example of cornea, right? So if you have cornea is a very immune privileged environment, because you cannot have the blood cells, because if you have blood cells or blood in blood vases in the cornea, it wouldn't be transparent. So in the beginning, you have some immune cells as keratocytes and macrophages that are special ones that are living there in the cornea. They are resident things. They are inactive, right? And when you have a deeper thing in the cornea, then what you have is you breach the epithelium, you go to the stroma, and that starts really calling pro-inflammatory cytokines production that calls macrophages, the white cells, to the cornea, and to prevent, even to reconstruct, even if there is no pathogens, right? There is no microbe infection, no infection. Just because you have to reconstruct everything, you have to call all these cells. Yes, and then it is something very complicated. We try to do something very, very simple, and we arrived with 22 differential equations coupled, and yeah, it's working. But I mean, but this is the result of piling up natural selection decisions for billions of years. right?
Yeah.
That's what happens. Yeah.
That's all very
There's
nice.
no simple things in biology.
Yeah. It's always a mess. Yeah.
Yeah.
Yeah. But that's nice. That's very nice. If there are no further questions, I know that there is a message from Camilo at the chat, and he says that he has great. I mean, first he congratulated your presentation and work, and then said that he has interest in digital twins, diabetes, modeling transport, and health in cities, and he got great ideas from your presentation.
Yeah, and if he want to discuss further, I'm available. It's just sending me an email.
Yeah, yeah. That's great. If there are no further questions, I'd like to thank you, Rita, once again. It was great. Thank you so
Thank you
much.
very much. I thank you. Thank you.
And yeah, with that, we finish. I don't know if Silvina would like to say anything.
No, I guess we are going to have our last colloquium 1 month, I mean, in a month from now, but we haven't organized it yet.
Last of the year.
Okay.
Yeah, last of the year. And hopefully next year we will have many more other things than besides the colloquium. Yeah. Many other things. Okay, thank you, Rita. Beautiful talk and work.
Thank you. Bye.
Thank you, everyone. Bye-bye.
Thank you. Bye.